Now, the structure of transfer RNA or tRNA. The nucleotide sequence, that's a primary structure of tRNA, was first worked out by Holy et al. In 1965 for East alanine tRNA. Since then, the sequence of about 75 different tRNAs ranging from bacteria to mammals has been established. The different tRNAs are all minor variants of the same basic type of structure. Several models of the secondary structure of tRNA have been proposed, and of these, the Cloverleaf model of Holy is the most widely accepted. 4 different regions of double-stranded RNA are formed during the folding process. See the figure on screen. Now, let us discuss about the Cloverleaf model of the tRNA. Amino acid acceptor arm. It is made by pairing of the 5' end, which almost always ends in G and is phosphorylated, and the 3' end, which ends in CCAOH. The amino acid is bound to the 3' hydroxyl group of the adenosine at the free 3' end of the acceptor stem. The amino acid is attached to one end, and the other end consists of 3 anticodon nucleotides. The anticodon pairs with the codon in messenger RNA, that is mRNA, ensuring that the correct amino acid is incorporated into the growing polypeptide chain. The next one is the anticodon arm. The part of the tRNA that participates in this complementary interaction with the codon of the mRNA is a stretch of 3 sequential nucleotides called the anticodon, which is located in the middle loop of the tRNA molecule. See the figure. Anticodon arm consists of 7 bases with the 3 anticodon bases in the middle. The anticodon is always preceded on the 5' side by 2 pyrimidines and followed by a modified purine. The other 2 loops are named after modified bases. The T-Si loop contains Si, which stands for pseudouracil, and the D loop or DHU loop has D for dihydrouracil. T-Si-C loop got a ribothymidylate residue, T a pseudouracilate residue, and a sidylate residue in that order. These strange bases are required for proper folding and operation of the tRNA. The T-Si loop and DHU loop are needed for binding to the ribosome and other protein factors involved in translation. Now, this is the table showing different RNA types and their functions. Types of RNA and primary functions. First one is mRNA or messenger RNA. It helps in translation, that is protein synthesis, and regulatory in nature. Second is rRNA or ribosomal RNA, and its function is to help translation, that is catalytic in nature. tRNA, transfer RNA, and it also helps in translation or protein synthesis. Now, hnRNA or heterogeneous nuclear, and its function is to precursors and intermediate of mature mRNAs and other RNAs. Small cytoplasmic RNA, that is scRNA, signal recognition particle SRP or tRNA processing, and this is catalytic in nature. snRNA or small nuclear RNA or snORNA, that is small nucleolar RNA. It helps in mRNA processing, poly A addition, it is catalytic in nature, and small nucleolar RNA, rRNA processing, maturation or methylation. Now, the regulatory RNAs, that is siRNA, miRNA, etc., it regulates the transcription and translation.